I'm pet sitting for a friend, and he has a lot of really cool books. This one is The Neuroscience of Human Relationships, second edition, Attachment and the Developing Social Brain by Louis Cozzolino. I'll read a little bit of it for you guys. Acknowledgements. It takes a village to write a book, an odd and dispersed sort of village, but a village nonetheless. I would first like to thank the village chief, my editor, Deborah Malmood, and her tribe at Norton Professional Books for their help, guidance, and patience through the birthing processes. I would especially like to thank Andrea Costello Dawson, who, with forbearance and a kind heart, was always available to offer advice and good counsel. And finally, thanks to Sophie Hagen, who has taken up the reins with intelligence and grace through the birth of the second edition. Many people at Pepperdine University, friends and colleagues, provided support and encouragement for this project. My thanks go to Robin Auger, John Baker, Bob DeMeo, Ed Shafranske, Tyler Vu, and Margaret Weber, day by day, year in and out, for their countless contributions to my work. I gratefully acknowledge the help of my research assistants, Melissa Flanagan, Olivia Hannon, Sharon Hiros, Maura Sauls, Drew Schontroop, Susan Sprocke, Natha Trita Savit, and James Wiley for their diligence, dedication, and hard work on projects interesting, taxing, inspiring, and downright boring. This manuscript also benefited greatly from the editorial artistry of Bruce Singer, Shannon Calday, and Luis Moricolith Latham. Many thanks to Linda Hudson and Christopher Warner of the Otis School of Design for their help in thinking through the drawings used in this book, and to their students, artists Nathan Frizzell and Modoro Kamada, who did excellent work in taking our ideas, mixing in their own, and bringing the drawings to fruition. I would also like to thank Carol Ermi for their use of her short story, and doctors Hiromi Kobayashi and Paul Wallen for images derived from their studies on eye morphology and amygdala activation. My respect and appreciation to my teachers, colleagues, and friends, Alex Caldwell, Lennart Heimer, Francine Inbinder, Zeb Little, Brendan Maher, Michael McGuire, David Meltzer, Hans Miller, Joe Palombo, Arnold Scheibel, Alan Shore, John Schumann, Renee Schwartz, Sandy Shapiro, John Watkins, and Jeff Weinberg for contributing knowledge, inspiration, and wisdom in all combinations. Many thanks to my friend Daniel Siegel, whose tireless dedication to me, the field of interpersonal neurobiology, and living life serves as a constant source of encouragement and inspiration in my own life. In the same spirit, I would like to thank my friend John Wynn, whose love, intelligence, and limitless enthusiasm for learning have become a central part of my own inner world. Both Dan and John dedicated considerable time to this manuscript, providing valuable editorial and conceptual feedback during the entire creative process. Special thanks go to Michelle Walker for her diligence and dedication to this project over the past 2 years. There is no way to measure the value of working with someone who is able to tolerate the complexities of my obsessive neuroses and the general absurdities of daily life with enthusiasm, good humor, and human caring. It's a mitzvah. Preface to the second edition. Since the publication of the first edition, the field of social neuroscience has grown at a mind-numbing pace. On the one hand, technical advances provide more windows into our inner neural universe. On other terms, like attachment, empathy, compassion, and mindfulness, regularly appear in scientific literature and highlight the deepening appreciation for the essential interdependence of the brain and mind. More and more parents, teachers, and therapists are asking how brains develop, grow, connect, learn, and heal. The challenge of for the first edition was to seek out and discover significant and relevant research. The challenge for the second edition was to organize an abundance of new scientific riches. A number of interesting developments have emerged between the two editions. Our understanding of mirror neurons and their significance to human relationships has continued to expand and deepen. As predicted in the first edition, the insula and cingulate cortices have gained increasing focus as researchers have turned their attention to mind-body connectivity and the importance of the subjective experience. Another important development has been the gradual shift in focus from the individual brain structures to functional neural systems, an important and necessary step forward. Exploration of what has alternatively been called the default mode network and cortical midline structures is providing a new window into the exploration of the self. Something that is growing increasingly apparent is that our attention to and analysis of others seems to have appeared earlier in our evolutionary history than self-awareness. Human self-awareness appears to be an emergent function arising from a combination of relationships, our ability to symbolize, and tools provided to us by culture. A great deal of neural overlap has been discovered in brain activation when we are thinking about others and ourselves. This raises many questions, including how we come to know others and whether the notion of an individual self is anything more than an evolutionary strategy to support our interconnection. All scientific journeys seem to lead us back to the old adage: the more we learn, the more we realize how much there is yet to learn. Fasten your seatbelts, hold on, and enjoy the ride. Luis Cozzolino, Los Angeles, California, January 2013. Introduction. I, me, mine. The miracle is that the universe created a part of itself to study the rest of it, and that this part, in studying itself, finds the rest of the universe in its own inner realities. Humans exist within a paradox. We conceive of ourselves as individuals, yet spend our lives embedded in relationships that build, shape, and influence our brains. Despite our fundamental social nature, the operative term remains I. I have relationships. I make plans with friends. I keep in touch with family. Our sense of reality is grounded in the experience of the separate self. It is from this perspective that Western science explores the brain. Yet while we are busy cherishing our individuality, our brains and minds are being stimulated, influenced, and regulated by those around us. Gradually, we are discovering that we are social creatures with brains and minds that are part of larger organisms called families, communities, and cultures. This awareness is making it increasingly clear that to understand a person, we need to look beyond the individual. Half a century ago, these insights led family systems theorists to shift the focus from psychotherapy from the individual patient to the family unit. The symptoms of the identified patient were thereby reinterpreted as a byproduct of the family's struggle for homeostasis. But is the family the best frame of reference from which to understand the human experience? Should we zoom even further out to tribes or cultures, or zoom back into an individual's biochemistry to get the best picture? If we use Mother Nature as a guide, we see that when she likes an idea, she sticks with it by conserving structures and strategies through increasing layers of complexity. Assuming this is true, which I do, we stand to learn a great deal from zooming in and out, from neurons to neighborhoods, while resisting the urge to become attached to any particular frame of reference. In this way, we may gain a deeper understanding of the interwoven tapestry of the biological, psychological, and social processes that constitute human life. Look closely at the body, and you will discover layer upon layer of highly complex interlocking systems. As you examine each layer, you will discover countless individual cells that differentiate and migrate to specific locations throughout the body. These cells, in turn, grow into an infinite variety of forms, organize into functional systems, integrate with other systems, and ultimately create an individual. This process we accept easily, but what about the notion that nature used the same strategy to connect individual animals, humans, into larger biological systems, which we call families, tribes, and species? Individual neurons are separated by small gaps called synapses. These synapses are not empty spaces by any means, rather, they are inhabited by a variety of chemical substances engaging in complex interactions that result in synaptic transmission. It is this synaptic transmission that stimulates each neuron to survive, grow, and be sculpted by experience. In fact, the activity within synapses is at least as important as what takes place within the neurons themselves. We know that neurons activate and influence one another through multiple biochemical messengers. Over vast expanses of evolutionary time, synaptic transmission has grown ever more intricate to meet the needs of an increasingly complex brain. When it comes right down to it, doesn't communication between people consist of the same basic building blocks? When we smile, wave, and say hello, these behaviors are sent through the space between us. These messages are received by our senses and converted into electrical and chemical signals within our nervous systems. These internal signals generate chemical changes, electrical activation, and new behaviors that in turn transmit messages back across the social synapse. The social synapse is the space in between us, a space filled with seen and unseen messages and the medium through which we are combined into larger organisms such as families, tribes, societies, and the human species as a whole. Because our experience as individual selves is lived at the border of this synapse, and because so much communication occurs below conscious awareness, this linkage is mostly invisible to us. Much of this book focuses on the unveiling of the social synapse and exploring some of its mechanisms. Through this exploration, we will examine how people, like neurons, excite, interconnect, and link together to create relationships. If you can accept the idea of a social synapse, perhaps you might consider a second theoretical leap. Neurons have three sequential levels of information exchanges that are called first, second, and third messenger systems. They are 1, the communication across the synapse, that 2, changes the internal biochemistry of the cell, which in turn 3, activates mRNA messenger ribonucleic acid, the material that translates protein into a new brain structure, and protein synthesis to change cellular structure. It is through these processes that the brain changes in response to experience. These 3 levels of information exchange are also taking place between individuals. In other words, when we interact, we are impacting each other's internal biological state and influencing the long-term construction of each other's brains. This, in essence, is how love becomes flesh. The discovery of the brain. Honestly, when was the last time you thought about your brain? Except for the occasional headache that feels like it's in the brain, we don't even remember we have brains. In essence, a well-functioning brain is invisible, which may account for why, at this point in our history, we know more about the movement of the planets than the workings of our brains. In fact, not too long ago, anatomists and physicians thought that the seat of consciousness resided in the heart, with the brain serving as the body's air conditioner, cooling the blood as it passed through. The brain's deeper significance is infinitely more complex than anything scientists have ever encountered. Studying the brain is like exploring a vast and ancient country with diverse landscapes, cultures, and customs. Our brains defy reduction to simple cause and effect relationships and anatomical boundaries. Like complex cultures, many of the interactions are subtle to the point of invisibility. To understand a culture, you need to be immersed in it, and to stay alert for small clues that may carry deep significance. Even a subtle diversion of gaze in the town square can tell the story of 2 people or 2 nations. Adding to this complexity is the fact that each of our brains is a unique interplay of our convoluted evolutionary history and the millions of personal experiences that shape it throughout life. Our brains are built in the enigmatic interface between experience and genetics, where nature and nurture become one. Crabb and Phillips, 2003, Ledoux, 2003. At first, genes serve as a template to organize the brain and trigger critical and sensitive periods. Later, they orchestrate the ongoing transcription of experience into genetic material. Through the biochemical alchemy of template and transcription genetics, experience becomes flesh, attachment takes material form, and culture is passed through a group and carried forward through time. Like every living system, from single neurons to complex ecosystems, the brain depends on interactions with others for survival. Each brain is dependent on the scaffolding of caretakers and loved ones for its growth and well being. So we begin with what we know. The brain is a social organ of adaptation built through interactions with others. To write the story of this journey, we must begin our guidebook with the understanding that there are no single human brains. Brains only exist within networks of other brains. The fact that the brain is such a highly specialized social organ of adaptation is both good news and bad news. The good news is that even if unexpected challenges emerge, our brains have a greater chance to adapt and survive. When good enough parenting combines with good enough genetic programming, our brains are shaped in ways that benefit us throughout life. And the bad news? We are just as capable of adapting to unhealthy environments and pathological caretakers. The resulting adaptations may help us to survive a traumatic childhood, but impede a healthy development later in life. The family is the primary environment to which our young brains adapt, and our parents' unconscious minds are our first reality, because the first few years of life are a period of exuberant brain development. Early experience has a disproportionate impact on the development of neural systems. In this way, early negative interpersonal experiences become a primary source of the symptoms for which people seek relief in psychotherapy. Interpersonal neurobiology. Interpersonal neurobiology, along with social neuroscience, Adolph's 2003, affective neuroscience, Panskep 1998, and sociophysiology, Gardner 1997, Alder 2002, are among the emerging fields of study at attempting to bridge the gap between the biological and social sciences. They all share the goal of understanding individuals within the context of the relationships into which we are born, in which we develop, and through which we live our lives. Interpersonal biology assumes that the brain is a social organ built via experience. Through interdisciplinary exploration, it seeks to discover the workings of experience-dependent plasticity, or the ways in which the brain is constructed by experience. At the core of interpersonal neurobiology is a focus of the neural systems that organize attachment, emotion, attunement, and social communication. The bidirectional causality between neural structure and experience requires a continual shift and focus from the brain to social behavior and back again to the brain. The social construction of the brain and the role of attachment relationships are particularly important in interpersonal neurobiology, as is the implication of scientific data to parenting, psychotherapy, and education. In addition to data from neuroscience and psychology, interpersonal neurobiology utilizes research from psychoanalysts, ethology, comparative anatomy, genetics, and evolution. In examining some of the processes that take place with the social synapse, we will explore a wide range of other fields that study narratives, storytelling, eye contact, touch, attachment patterns, and body language. Thus far, those of us interested in interpersonal neurobiology are primarily psychotherapists and educators attempting to utilize neuroscientific data to inform and improve our work. Cozzolino, 2010, Shore, 1994, Siegel, 2012. Like curious toddlers, we are full of questions, such as which neural networks compose the social brain? How is the brain built and rebuilt by relationships? How do brains regulate one another during moment-to-moment interactions? How do parents, therapists, and educators activate and guide neuroplastic processes? What are the effects of isolation, stress, and trauma on the social brain? Caregiver nurturance sets us on a course of physical and physiological health, or when it is lacking physical and mental illness. Because of the causal link between interpersonal experiences and biological growth, We are particularly interested in the impact of these early caretaking relationships when the neural infrastructure of the social brain is forming. We also know that the brain is capable of change at any time, and that social interactions are a primary source of brain regulation, growth, and health. Those of us who study interpersonal neurobiology believe, and research supports, that friendships, marriage, psychotherapy, in fact, any meaningful relationship, can reactivate neuroplastic processes and actually alter the structures and biochemistry of the brain. In thinking about these processes, a multitude of questions arise. How does therapy work? What are the best ways for parents to help build their baby's brain? Why do some people seem to have no conscience, whereas others are crippled with shame and guilt? Why do some find cues to abandonment in almost every human interaction? How does the brain stay healthy, and how can we regain mental health after we fall ill? Parents, educators, and therapists, those of us who should be most concerned with shaping minds, have traditionally paid little attention to the brain. I've heard therapists say that psychotherapy is an art and that the brain is irrelevant to their work. I would respond, as with any art, that a thorough knowledge of our materials and methods can only enhance our skills and capabilities. The brain is a treasure trove of information about where we have come from, what we are capable of, and why we act as we do. It holds many secrets about how we can know ourselves better and improve the way we do psychotherapy, teach, and parent our children. As a therapist, I am especially interested in how relationships reshape the brain throughout life. Tens of thousands of hours of interacting with clients have provided me with an intuitive sense of how and why therapy works. I've watched as my focused attention, consistency, and caring have been taken in like water at a desert oasis. I've experienced the gradual building of confidence and strength as my presence has been integrated and used as a source of security, guidance, and emotional safety. I've also experienced how working with my clients has changed me, inspired me, and helped me to grow. It is the power of being with others that shapes our brain. Exploring how these insights apply to therapy and everyday life is one of the most exciting aspects of the work that I do as a therapist. And it is what has inspired me to share my ideas about the brain and the mind with you. Well, that was the intro. That was pretty long. I'm going to take a break. I'll probably read this one more another time. But yeah, that is the Neuroscience of Human Relationships, the second edition by Luis Cozzolino, and I believe it was written in 2013. If you guys want to check it out.